Now that we've learned the individual amino acids, it's time to stitch them together to make the peptide bond. And this is ultimately how proteins are made, by attaching many amino acids together. So this is a particular reaction that occurs when the amino group of one amino acid reacts with the carboxyl group of another. And they're going to form this new functional group called the amide, or the amide. I will pronounce this amide. This is a reaction type that we're going to see over and over in this course, called a condensation reaction. And so condensation, if you think about what condensation is, like if you're driving in your car in the wintertime and the inside of the glass gets all fogged up, right? That's water vapor that sort of then deposits or condenses onto the window, and it's water. So anytime we're talking about a condensation reaction, we can say that it's going to form water. So a condensation reaction forms water as a product. So that's always going to be a clue there for what's happening there. So let's take two amino acids. Here we're just saying R1 and R2 for the side chains, but this is a little different representation than what we've been looking at. It's not a Fisher projection because the carbon skeleton's not running vertically. These are just a regular old Lewis structures, so we're not really expressing anything about the 3D nature of these. We're just using them as Lewis structures to highlight the chemistry that's going to go on here. But notice again, I've got the nitrogen, so N, this is my alpha carbon, remember? That's what we call the alpha carbon. That's the alpha carbon. And then we have the C double bond O here, right? So it's a carboxyl group. Same on the left hand, the R2 amino acid. I've got the N, I've got C alpha, and then my carboxyl group. And so we can stitch these together to make a peptide bond. And the way this happens, I'm going to use an arrow to represent a reaction. We kind of did this when we talked about acid-base chemistry a few weeks ago, but I'm going to use an arrow to show a new bond that's going to form. So I'm going to take an arrow from somewhere near this nitrogen, and I'm going to point it toward where that carbonyl carbon is. And so when I'm showing that, when I show a line like this, I'm going to call that the new bond, right? I'm going to make a new bond, and I'm using that colored arrow to demonstrate that. So that's a new one. Well, remember, carbon always needs 4 bonds. We will never see it with 3 for what we're doing, and it never can have 5. And so that means something's going to have to give here. So I'm going to demonstrate that a different bond is breaking by drawing a line sort of through the bond and then kind of pointing an arrow onto that oxygen. What I'm really showing here is how the electrons are moving around, forming bonds and breaking bonds. And so here, this is the old bond is broken. In that case is what we're demonstrating. And then the rest of the atoms here, just look at this carefully. I've got an oxygen that's going to go away, and I've got 2 hydrogens here that are going to go away. And so I'm going to circle those, right? And they're going to end up, those 3 atoms are going to end up in the form of that water down here, right? That is why it's a condensation reaction. And then there's going to be a new bond between the nitrogen of the second amino acid and the carbonyl carbon here. And so in the product structure down below, again, here's the one on the left and the one on the right, I've got this new bond between the nitrogen and the carbon right here. And so I'm going to draw that one in this color here, since that's the color of my arrow. So that's our new bond right there. This is called the peptide bond. It is the bond that joins together 2 amino acids covalently. Now, this is a new functional group, like I said up here. It's the amide functional group. And amide is when we have a C double bond O, and then that carbon is bonded to a nitrogen. And so it's kind of the cousin of the carboxylic acid in some ways, in that if this were C double bond O, we would have a carboxylic acid or a different functional group called an ester that we'll learn later on. But anytime we see C double bond O, then bond N like this, it's an amide. This can be in the middle of a protein like this as part of a peptide bond. It can also be the side chain of asparagine and glutamine as we see in our notes. So we're going to add this new functional group to our list of groups here. So just a couple other things about this. I will go ahead and label that, right? That is the peptide bond. That new bond that we formed. And because if you look at this dipeptide is what we would call this, it has on one end essentially an unreacted amine, and on the other end an unreacted carboxylate. So if I look on one end of the molecule, it's different than if I look on the other end of the molecule. In fact, this has what we'd call a polarity, that the R1 amino acid, whatever it happens to be, serine or asparagine, whatever it is, that it is what we would call the N-terminal residue. This is like, think of like a train tracks, right? The terminus is where things stop. And then similarly on the right-hand side here, I've got R2. So whatever amino acid that happens to be, let's say it's histidine, we would say that histidine, or R2 then, is the C-terminal residue. So I'm going to label those on here, that this is the N-terminus. And that this is the C-terminus. And we could use that as an adjective to say C-terminal residue and terminal residue. And so I've been using this term peptide. This is just a generic term that we use to describe, some number of amino acids stitched together. And if it's just a couple or 3 or 4, we might say, dipeptide, tripeptide, that sort of thing. But eventually you get to the point where it's just too many, and we would just call it a polypeptide, right? Polypeptides then fold up and become proteins, essentially. And I'm going to use the term residue from now on when talking about an amino acid that is part of a peptide system like this. Because R1 up here, right, was an acid, it had a carboxylate group there. But once it's formed this peptide bond in the amide functional group, it's no longer really an acid. Yeah, the right-hand side here is an acid, but this group here, there's no acid here. And so it's really not true to call it an acid anymore. That's why we really call it a residue. So I'll use that term. When I say residue, I'm referring to an amino acid that's been incorporated into a polypeptide. And as I just mentioned, the ends of the amino acid give us the termini, N-terminus and C-terminus. And it's important to recognize, which is which. We typically read these these polypeptides from the N-terminus to the C-terminus in that order. And we'll see that as we move on to some other things. So a couple other properties about the peptide bond. You might remember the term resonance, and this is when electrons can move around. And so what we're kind of showing here in this figure on the right is that the electrons that were hanging out in this lone pair, that nitrogen, they can kind of pop down here to form kind of a double bond between the carbon and the nitrogen. And then again, carbon then would say, hey, I can't do that. That'd be 5 bonds. Carbon never makes 5 bonds. And so it can kind of relieve that stress by moving these electrons, just one of the bonds from the double bond, up onto that oxygen. And so that movement of electrons is called can lead us to this other resonance structure down here, where we have a little bit of double bond character on the peptide bond. So this is true for all amides, that they can have these 2 resonance forms. And later on in the course, when you do this in lab, the peptides lab, you'll explore this a little bit. But we use this kind of cool double-headed arrow to represent that we've got these resonance forms. And really what's happening is the electrons are kind of smushing all around back and forth here, and kind of the true structure is kind of an average of all of these. Now, remember, double bonds cannot rotate. So there's no rotation around this bond right here, right? The double bond kind of locks these in and disallows any sort of rotation. And remember before, we talked about conformational isomers or conformers, that when you rotate bonds, you can kind of end up with the same molecule, just in a different orientation. But because this bond can't rotate, it means that we can get some distinct isomers called cis and trans isomers. So we're going to use this term to demonstrate the way that atoms are arranged around a double bond. So for example, we could have a carbon-carbon double bond. I'm going to show it just as two lines like that. And I'm just going to say that they're, whatever the R groups are, it's unimportant, but they could look like this. And if it's in that orientation, right, where the two R groups, the big bulky groups, whatever they happen to be, are on the same side of that double bond, we call this the cis configuration. Alternatively, if we had something like this, where one of the groups was on one side and the other group was on the other side, we would call that the trans configuration. These are isomers of each other because I can't convert between the cis and the trans without kind of breaking one of these bonds and then allowing a rotation. Remember, that was kind of the differentiation between a conformer, where you can just rotate bonds around or rotate the molecule around, and an isomer. If we're going between isomers, we have to break a bond. So if I had a model kit, you'd hear a little pop of bonds breaking and making in order to interconvert these. And so it turns out that in most cases, the peptide bond will be in a trans configuration, and you can sort of see how it spaces things out. It spreads groups out a little bit more. And so a lot of times in chemistry, not always, but a lot of times spreading things out in this trans configuration makes things a little bit more stable.